Good morning, ladies and gentlemen. My name is Anna Vogtman, and today I have a pleasure to replace my colleagues who had to go back to ESA today and invite you to this very interesting, hopefully an inspiring discussion about topics that ESA usually does not pick up that often. Hibernation. It sounds like a sci-fi to many people. However, today I hope we will be able to learn whether it's really sci-fi or how close we are to application, whether it's realistic to actually have hibernation being applied into operations, spaceflight operations, to support our astronauts on their space endeavor. So today's session, we will give few presentations. So first I will start to tell you more about programmatic achievements from ESA side. Then I will pass it by to my colleagues, Matteo ESA-HRE, who will give you an overview about what hibernation is. Then we will pass it by to Dominique Moser, who will tell you more about the mechanistic background of hibernation and torpor. And finally, we'll talk about the application to radiation research. So let me start with the programmatic. Some time ago at ESA, we did a so-called feasibility study on hibernation. It was from ESA side, it was the first attempt to assess whether hibernation would be actually a thing that we could apply in our spaceflight operations. However, there were studies before, and this feasibility study was using or summarizing everything that was done until date. This study, the report was published in 2019, so it's a bit of time. So I'm later on, I'm very keen to learn from our experts about the advancement in this field, and few things have been published that I'm very curious to ask you about. But thus summarizes and thus assesses the system-level requirements or identified system-level requirements that are needed to be fulfilled in order to apply hibernation in spaceflight. So let me just quickly summarize the key finding of that report. Actually, to my surprise surprise, this report actually did not brought that knowledge closer to our application. So it showed that actually it is very feasible and realistic to achieve that level of technology to bring those resources that are needed to space in order to sustain hibernation. They did look at the system level requirements, they look at the architecture of the spacecraft, at the budget mass, that was very realistic. They also looked at the crew requirements. So whether we need to have a specifically trained crews on board to actually be able to induce hibernation, or whether the usual medical training dedicated to a specific profile of the mission would be sufficient to support hibernation activities. And also in this report, they identified the AI developments that need to be developed in order to apply those kind of tools into spaceflight operation. But except identifying the system and human level requirements, the report also identified research gaps in order to achieve. So there were long and short-term milestones where certain gaps to be addressed by our research program were addressed. They created this roadmap, and I will invite you to investigate that roadmap in the outcomes of our topical team study that was running for quite some time, and that Marco Cherry would talk about a bit. So in that, and I also encourage you to look at our webpage. You will find there like overview. It's a really nice movie overview about the hibernation study that I just briefly mentioned. So with this, I would like to thank you. I also would like to point out that we will get through those presentations, and then we will start a discussion panel where you have also an opportunity to ask questions to the panelists, but please bear with us until the end of all the presentations. So with that, let me invite Marco Cherry to the stage. Marco, the stage is yours, and please take your time and enjoy your presentation.
Thank you, Hannah. That was very kind of you. especially because now you have a dear friends in Frankfurt, which many of you may know is Marco Durante, and I'm Matteo ESA-HRE, so we always share a name. Yeah, Marco and Matteo, that's fine. No, you know. I must tell him, but I don't know if he's going to be happy to be sharing name. No, I found it funny. Okay, so thank you everyone for inviting me, for having me over. And yes, Hibernation is a. I'm gonna go this way. Right. Oh, okay. Oh yeah. I have another name. I'm I'm also Dominic at the moment. So careful, because if you shout your friend's name, I may turn up. But it's a good way to make new friends, right? When you go to a conference. So, well, in the meanwhile, yes, Hannah was correct. So ESA had, I would say, the long side to investigate hibernation as a possible field of investigation for developing new countermeasurement, and not only for space. Actually, there is a lot of health research involved, of course. But the space is more exciting, I have to say, especially for scientists. And your colleague will look at you with respect when you say, I do something with space. So, well, you must be a rocket scientist. I say, not really. But anyhow, hibernation. So what is it? Let's see how it goes. Like this. Forward. Okay. So as I say, ESA had the idea to establish a topical team on the topic, which I say is really visionary at the time. Because when it started, hibernation was mostly a field of ethology. So people were going in the field and look at animals, were entering this Winterschlaf in German. And I don't know if my pronunciation is correct. Great, thank you. So stopping by for a long time, and for quite some time, people had the idea that they must be useful. They could be an interesting application. But it was mostly around going around the world and see which animal did it for how long, where, and setting the stage for the investigation. There was no much physiology, a little, but not very much. And especially no one there at the application at the time. So topical team was established, and I was a member of the first one, and it got renewed, and kindly I was offered the chairmanship by Jürgen Berheiterhain, which was the previous chair, and we all will enjoy his guidance, was unpayable. So where we go from here? You saw hibernation is sci-fi. Now, because I'm sure you may be familiar with this kind of imaging, and you may test your Hollywood knowledge of blockbuster movie, try to identify what movie is this taken from. I can spoil the secret at the end if you're interested. But that's not what hibernation is. So there is no freezing, right? That's cryonics, it's a very different world. And the director takes some liberty in doing this kind of scene. Most of the time, people are naked in hibernation. It's unclear why. I mean, it's clear for the screen, but not really for scientific reasons. If there is a director among you, raise your hand and we can give you advice. All right, but so what is truly hibernation? It's something like this. This is a marmot from the Alps, and yeah, all right. And this is the body temperature of this animal, which is, as every mammal, is 37. But what happened? See, the body temperature suddenly dropped to about 10 degrees. This is ambient temperature, about 8 degrees. So it's almost a passive element of the environment, like was a rock there. And for a long time, people have thought that this was the event. Why? The event we were interested. Why? Because if you lower body temperature, if you lower temperature overall, you slow down the speed of chemical reaction, and therefore you slow down biological time. Not just biological every time, but especially in biology. To Gerhard Hellmeyer, which is also a father of the field, he showed that the event is this one. So metabolic rate here, which is how much oxygen this animal uses, how much energy is using to stay alive, to act and behave, suddenly drop about 95%, and therefore the animal cools down. So it's the reverse of what we were thinking. First idea, we cool down and therefore save energy. No, we save energy and therefore we cool down, which totally reverses the angle we're looking at this event, and provides some interesting clinical insight. So, moving on this idea, I'd like to show you how it looks like in, I don't know, real life. Those are 2 mice from my lab, and the bottom one is a control, so it won't go into metabolic depression. Technical word is torpor. I'm going to use this word from now on. The one on the top will, and is really accelerated because I have, say you have 15 minutes, so I have to cramp up a movie in 30 seconds. So you see, it doesn't really move like this, a mouse. But the one on the top, you see, is kind of turning off. This is a infrared thermal imaging, and slowly go back to ambient temperature, pretty much like the marmot. What happened to this mouse? He was put in a condition in which he could choose to enter torpor. So spontaneously, it does that. In lab, they do that. Actually, she does that because female mice are more prone to enter torpor. And therefore suspend their activity and stay there for, mice can stay there for up to 24 hours, more or less. But coming back to the original question, so why are we interested in this kind of things? Well, because during torpor and during hibernation, there is a very, very weird physiology. So the body and the way it regulates and works change drastically compared to what we're used to think. So every organ and every system has to readapt to a low energy consumption function. So meaning there is no, the amount of money that were going out in town to keep the business open, they're not going out anymore. So business has to close and survive. And the same thing has to do organs in our body. So lungs, liver, heart, brain, bones. Every organ has to obey a specific command, which is consume as little as possible. Just the minimum bearable amount to stay there and don't die. And that's especially important because animals in torpor and hibernation don't die. The heart will still beat, the lungs will still breathe, but very, very, very slow, at the slowest possible level. It's like if you were going and touch the wall of death, but stay on the life side. And we'll see how this happens in interesting cases. And if we have to list all the interesting changes, there are plenty of, as I mentioned, there's an open field of research. Every tissue and organ is open to investigation. I just highlight a few, but I was pleased to see yesterday a fellow physician mentioning the hemostasis hibernation study, in which it was shown that bears don't develop, they're protect against thrombosis during hibernation, because that's another interesting application that came out from hibernation study. And as I mentioned, a few interesting facts. For instance, there is no urine production and no waste production for months, right? So the kidney pretty much works in a very different way. There is a reduced metabolism, so you don't eat and drink and for months. Consciousness is, let's say, very much reduced. And the brain completely, or at least tell neurons not to talk to each other anymore. Synapses get reabsorbed and firing rate goes down incredibly. After all, you don't need the brain to stay in torpor because there's nothing you're doing. So it's a totally wasteful organ at the time. And so it gets put on pause. GI tract, of course, you don't eat, so it changed the leaking and the permeability and the microbiota completely disrupted because since you're not eating and you're at a low temperature, all the population of microbes are changing. Very interesting for space application, you preserve muscle strength. So animals that stayed, say, with no moving for months, 9 months, when they wake up, they are perfectly able to move and don't lose muscle strength. And there is a lot of investigation in this, and the same goes for bones and maintaining mineralization and structure. And that makes sense. Otherwise, if you were waking up from hibernation and you were going to live your 3 or 4 months of life, because those animals live 3 or 4 months every year, you should be able to run right away and to go for eating, feeding, reproducing, whatever you need to do. So also, and I think Rainey at the end of the talk will talk more about that, especially interest, animal in torpor had a significant increase in protection from radiation at cell level. So that's a biological development for cell, which we are also looking deeply into it, and it's unclear why this happened. There were a few hypotheses were put up, and I don't want to steal Rainey's presentation, and I'll leave her all the time to go in, but we have hint for some surprise, especially from my interpretation personally, is that the change in gene expression for cell in torpor that is driven by the need to reduce energy consumption is also the one setting up a genetic stage that protect against, or at least allow the cell to have more time and more precision in repairing the damage. So the damage kind of happened anyhow, but you can this time turn it back on the machine and the cell doesn't die. We don't know yet this will give you cancer in 30 years, we might, but on the acute stage, cells survive. It's like if you were fixing your engine while the car was not moving. But if you have to fix your engine while the car is moving, that's very more complicated. So that's the overall idea. If you have more questions, of course, I'm happy to answer. Or the immune system, lymphocyte, all migrate back to lymph nodes. So you have a theoretically suppression of immune function, which doesn't, let's say, convey into increase in infection rate, is actually protect your body against self-damage from the immune system. So as I mentioned, very, very different way to survive. And all of this is interesting. And in a neuroscience conference and in physiology conference is always, say, generate a lot of questions. But here, the main question comes usually like this. Okay, cool. So we can send squirrel to Saturn, and then what we do? We can send bear in space, and so. So the main question comes down to, can human hibernate? Is human hibernation possible? Because this is the effect, okay? That translate from science to translational science. And so what I'm going to show you here is human cases in which something like this might have already happened. There is a lot of reason to sustain this thesis, and I'll try to convince you that that is true. So there is a lot of anecdotal history, right? And if we go back to the past, and also on the present, this is an interesting documentary on Netflix. You can watch it, Last Breath. A submarine worker, one that's worked on oil platform, had an accident, was cut off of oxygen supply, stayed there for almost an hour and survived with no oxygen. Just an interesting case report, and you may see, well, there are many of those. Every year there are cases like this. Even in history, this is the case of Hannah Bagelholm. I'm going to bore you with a little bit of history here, but she was sentenced to death in 1600 in London because she killed her two children. And she survived hanging in winter. And this is a xerography of the time. You see her family here. They tried to help her. She was hanged, and apparently she didn't die for the deceleration trauma. So her family was trying to help her. They were picking her up and shoot her up in high again, hopefully to kill her. So careful of your friends' help and your family help. No, well, I'm kidding, but once she looked like she died, she just, the law, according to the law at the time, they had to be hanged for at least an hour, something like that. So she looked like she was dead. She was carried to the cemetery and blah, blah, blah. And then suddenly someone realized she was still alive. And then they call the doctors and the judges and whatever. And the final judge decided that she was guilty as charged. Sentence was carried on. All good. And so she was let go. And she lived 13 years after surviving hanging in a very cold morning in London. This is the most famous case, Dr. Hannah Bagelholm. She survived accidental hypothermia, about 13.8 Celsius, possibly the coldest. She was named the coldest woman on Earth. That's not, probably is not true. There are reports of lower temperature, but in medical settings, let's say, we all measured the temperature and we all confirmed was that this is still the record, I guess. And she was cross-country skiing and went head front in the frozen lake. It took more than an hour to recover it. Her heart stopped beating, of course, and it took days to revive her. She revived, she gained function back. She's a doctor, she's a radiologist, and she's perfectly fine. The only complaint she has now, I think she has a little numbness in one of her finger that makes difficult for her to peel potatoes. That's the, but good thing. But those cases have something in common. People actually say they died, and they were revived somehow by something, or they didn't completely die. So let's go to something more interesting. 19100, British Medical Journal, anonymous report, human hibernation. Apparently in the city of Pskov in Russia, I realize just now not too far from here, a report for human hibernation was put down in a, I don't know who wrote it, the history of science would be interesting to find out, saying that in this town, the citizens, the peasants were all going together in a large house they built at the center of the village to pass winter together, so stay there for winter, but not to help each other for food and whatever. But they built a little cell, like in a monastery. They were going into those cells and they were sleeping the winter off. There is a Russian name for that, which I don't know if it's analog of Winterschlaf, is Lochka. If there is anyone who knows Russian, maybe you may correct my pronunciation. Go forward to a more interesting case, because those were all cases. I thought, well, she was in the 1600, who knows what was going on there. This is 9100, who knows. Oliver Sacks wrote an interesting peace report in one of his novels. This novel is a biography, we say. This is the collection it was taken from. It's the story of Uncle Toby. I'm going to make it short for you, but it's a very interesting and deep story. Uncle Toby was a person he met during a house visit in his early days of physicians, and they were in his house for different reasons. He just noticed this old person sitting there and doing nothing. So I said, well, who is this? This is Uncle Toby. And what does he do? Nothing. He stays there. I say, no, the family says 7 years ago, he froze. He stopped moving, stopped everything. And we feed him, we shave him, we take care of him, we dress him. But we put it on the chair, and then we put it on the bed, and we put it on the chair, we put it on the bed. So he suspected hypothyroidism, measured thyroid hormone level were under the 4. And they decided to treat the guy, and said with the family, so we could give him thyroid hormone, at the time was readily available. And they try, and he woke up, right? He woke up, and he didn't remember anything. The 7 years they were passed behind. And there will be an happy ending story, but it's not very happy, because together with Uncle Toby, a small cell lung cancer also woke up. And going back to radiography, fortunately taken a few months before freezing, lung cancer was already there. And so hypothesis, so those kind of cancer discern and produce a lot of neuroactive substances, a lot. There is a vast literature of effect induced by those cancer. And idea is that this specific tumor may have produced something very peculiar that may have reactivate torpor circuits in Uncle Toby, as Uncle Toby went into hibernation, and at the same time hibernation suppressed cancer cell growth. So the two organisms find a balance, cancer lived, Uncle Toby lived at the same time. And since hibernation is a, say, systemic phenomenon, when it's triggered, it suppressed thyroid hormone production. So what Oliver Sacks saw was the consequence of torpor, not the cause of torpor. And of course, if you give thyroid hormone, you waken up. And waking up the tumor as well, Uncle Toby died, unfortunately. But that's for us is an interesting story. And then there are even more interesting story, people coming back to life. So what is people coming back to life? This is a famous picture just before pandemic. This Mount Everest, for the one who knows, or of you who may have been there. See, it's kind of, there's a lot of people there. And it's like Monday morning in Bologna. But why there is a lot of people? Mount Everest became a touristic destination. It's still expensive to go there, but it's relatively easy in terms of the physical challenge, except for the fact that it's very, very high. So of course, technology help you and the guide help you, but people still die on Mount Everest, about 10%. So you can go with your 9 best friends, one will stay there. Choose carefully your friends. That's an opportunity they will never present themselves again. All right, so where people die. So you've seen on Mount Everest, Sherpas die on low altitude, meaning low, I mean below 7,000. And that's because they work, and that's mostly work accident. Tourists die on the top here, on the summit, right? Why they die over 8,000 meters? Because 8,000 meters is the death zone. So there is so little oxygen here that if we were to be teleported there now, we'll live about 20 minutes, more or less. If we were adapted, as mountaineers do, for a couple of months of 5,000, 6,000 meters, 7,000 meters, we could do here, but we couldn't survive for longer than 18 hours, 24 maybe. So the Camp 4 is here, 7,900 meters. And then from here, mountaineers summit Mount Everest, and they have to go up and down, as I said, within 18 hours, tops 24, no more. Otherwise, they die. So a lot of people died, and there are cadaver and bodies here, plenty, because no one goes there and bring them home. It's very hard. You can't really do it. That's a macabre story, but there is a body called Green Boot here, which is a reference point. Now, on the map, they tell you, walk ahead until you see green boots. And then green boots turn left. And there is this person which is dead, and you don't see his face, you just see the green boots. So it's a dangerous place, you mentioned. And many people died. And these two guys here, Lincoln Horn, Australian mountaineer, and Beckweather, an American, they, in 2005 and 1998, I think, they all did the same story when back there. They couldn't make it, so they were left. He had a friend in his crew, which was a cardiologist. He declared him dead. He was declared dead as well. Families got a phone call like the day after saying, sorry, madam, your husband's dead on Mount Everest and will never come back. So what happened? Well, those two guys, they weren't recovered as Anna Bagelholm and revived. Those came back on their own, on their feet. He walked back to main camp. So in the morning, after 26 hours later, 28 hours later, he walked to Camp 4 and say, I made it. I was happy to be alive. He was frozen. He had issue, but he was alive. And he was found 46 hours after he was left, sitting on the lotus position, meditation, looking at the Himalayan dawn coming up, by two English mountaineers that were going up Mount Everest. And famously, one of the famous sentences of history, he turned to them and said, I think you're surprised to find me here. I said, yes, we're very surprised. And they brought him back, and unfortunately, he died of cancer 10 years later, and he's still alive. So why I mention this? Because those are humans that didn't just survive cold. They woke up, and the only natural state that allows you to die and come back is torpor. There is no other explanation. Every possible condition, if they have brain edema, heart failure, whatever, would have stayed and would have just kill you, or you would need medical support and attention. They came back on their own, and that's the only thing that can help. And that's not a casual thing that is on Mount Everest, because the conditions on Mount Everest are very similar to the one that push some kind of animal into torpor. Fasting, because they don't eat over 7,000 meters, low oxygen suppress appetite, cold, and low oxygen. So a triple of condition that some animal, they call facultative heterotherm, like the lab mouse, they feel as dangerous environment, and they can activate this old system of defense, which is torpor, and they can do it. So our idea is that what happened, and if we calculate for if there are humans are able to enter torpor, how many? Is this true things, or is just a random accident? So on Mount Everest, there were 330 deaths in the last 100 years, all right? And this is a statistic we're taking, but as you can see, there is, of course, accidents and stuff, so we should take those deaths off. We are interested in that by exposure, which is around 30. So people who die because of the environment. And if those 30 cases died for exposure, and we have 2 cases of torpor, that means that about 10% of humans able to enter torpor, that we don't know about it. And this is interesting because we don't know if we can enter torpor. So assuming that this is true, and Lincoln Horn and Beckweather are still human able to enter torpor, they would have never find out unless they were there. And we don't know if we are able to enter torpor, because very rarely we found ourselves, I would say never, in a condition to activate this extreme defense mechanism, right? So for us, as in the topical team, we have much discussed about this and to trying to find a test, something that we could run, both in terms of molecular testing or behavioral testing, whatever kind of testing that could tell if some person is, let's say, prone, torpor prone, could be familiar entering torpor prone. And so it's something I encourage you to think about it. But there is more to support the idea that human can enter torpor. There is, of course, rare conditions are always a gold mine for people looking for weird effect. And especially in neurological conditions, there are more diseases for the brain than for the entire rest of the body. So you can find very, very weird syndrome as a neurologist. And this one is a very interesting event, syndrome. This is a temperature trace of a patient. You see, it's 5 days, continuous recording. Every morning at 6 a.m., this person's cool down and cool down and cool down and cool down. And if I didn't, when I found out this paper, which was published in 2002, I showed it to a friend in hibernation field and to trick him. And I said, look at this data. And I said, what is it? This is a hamster? That's what he said. But because it really looks like an hamster. The hamster do this in winter. Say, no, it's a human. So we start looking at other cases. And there are lots of different names for this syndrome. The most commonly used maybe is spontaneous periodic hypothermia or Shapiro syndrome in case there is also a small anatomical lesion in a specific part of the corpus callosum just over the perioptic area, which is not coincidence. Look for this syndrome. I found 103 cases in the last 120 years reported in the medical literature. They may be more, but those were the ones available. More or less distributed as this, so 20 in North America, 15 South, most in Europe, and some in Asia and Australia. How do they combine? So pretty much same for male and female. As I mentioned, in the natural world, female are display usually longer period of torpor and easier for female animal to enter torpor because testosterone is, let's say, an enemy of torpor. So male have a shorter torpor bouts and it's a little harder for them to enter torpor, but whatever is this condition, seems to work fine in both genders. And there is a small group in which circadianity is maintained, which means event happen the same time of the day, other not, and seasonality happen in a few cases also, so just in winter and not in the other area. There are two populations that display the syndrome: children and adults in their middle age, like around 40 and 50. I think I just passed the critical threshold for getting this disease, so luckily I'm done damn it. And this may be congenital. This may be induced somehow. Most of these people, at least a portion of these people, report to a flu-like infection something before that could feed into the idea of an old brain circuits that may be activated by some very rarely developed antibody or something immune-related. And body temperature drop, in most people a few degrees, 35, 33, but there are people that go to severe hypothermia and they survive for longer time. So they do actually enter torpor. And the duration about from hours to weeks in this condition, from daily to yearly, the frequency of the bout. And in adult, often, then the disease heals from itself. Somebody stays for his entire life. Doing clustering analysis, we have found out there are actually two syndromes, very, very, even more rare, but I'm not going to go into this detail. They're slightly different between the two. Interesting fact is we don't have any idea what triggers this syndrome. So clearly not genetics, something genetically in the environment, but it may be that we are possibly torpor prone. Maybe some rare kind of flu in the torpor prone population will trigger spontaneous periodic hypothermia. And I like to conclude my talk with some famous word. This is Dr. William Coley. Some of you may know it. He was the first who thought of immune therapy taking care of Mr. Freudenstein. So he has a cancer, then he developed an infection, and the cancer healed. And many will say, well, good, a miracle happened. He thought there may be something in the immune system that could be used against cancer. This is an anecdotal evidence, There were no statistical analysis on that, just case of one. But something happened, something that maybe replicate once we understand it. And then immune therapy came along years later. And I like to conclude that this hibernation is very much the same case. We have anecdotal evidence that support the case that human could enter torpor. We're working and investigating the mechanism. It's worthwhile to invest to understand the how to develop the translational part. If the data is not the plural of anecdote, as some of you may have heard from Raymond Wolfinger, a lovely and great American statistician, this is a wrong sentence, wrong quote. That's why I reported all the time, because what he actually said is right the opposite. I don't know why this fake news came in the web, but he said the plural of anecdote is data. And with this historical update on a famous figure, I'd like to thank you very much and for your attention.
Thank you, Matteo. Very nice for this really nice and thorough introduction. Now let us get into a bit more details about how torpor works. So please welcome Dominique Mosser, who will give you a presentation. Dominique, the floor is yours.
Thank you very much. So hello and welcome. I have to apologize very much for my voice, but I will try to do my best that you understand maybe a bit. So thank you very much also for coming here today and to invite me to this great workshop and to give us the possibility to present our work where we investigate the possibility of human torpor by cross-species transcriptome analysis. So what is the big goal? We heard it quite often in the last few days. It's to realize crewed missions to Mars. As we all know, Mars is quite far away and it also will take quite a while to get there. And based on all the knowledge we gained from research in space by now, we expect that the space travelers will be exposed to quite a lot of mental stress, deconditioning of several physiological systems, and also radiation is a point that should not be neglected, as we heard it several times. And there are also a lot of technical challenges, especially the high payloads that have to be taken on a spacecraft, just regarding the nutrition and the oxygen a crew would need for such a long time of journey. And it seems actually quite elegant to maybe put the crew in some kind of inactivity or a torpid state, and this is what we are working on. And already now, design facilities exist that aim to develop by computer modeling Mars mission scenarios with regard to human torpor. And they consider the mission design, habitat design, life support systems, and associated logistics, as Anna has mentioned before. This one concurrent design facility modeled a mission and came to the conclusion that the presumed metabolic reduction to achieve optimal conditions would be approximately 25% for a duration of 85% of the whole mission. And all these assumptions and calculations that have been made are based on the knowledge that was gained from experiments on bears. And based on these assumptions, also quite nice hibernacula were created. So you can see that they should consist of a very cozy soft shell individual pod, which will provide an optimal noise protection, personalized air conditioning, and illumination control. The astronauts will be connected to a medical interface to monitor health, and of course the whole pod will be shielded against ionizing radiation, and all processes will be monitored by artificial intelligence. And during non-torpid states, these pods can be used as private quarters. So far so good, looks nice, sounds nice. Maybe it will be achievable. But before we go into human torpor, we just have to reflect a bit. Where are we now? What do we know about torpor? So torpor is an adaptation to shortages by a state of metabolic depression. It is characterized by physical inactivity and very low energetical requirements and an endogenous reduction of the metabolic rate that is accompanied by a subsequent decrease of body temperature, cardiac output, and breathing rate. What we also know is that several processes are downregulated very strongly during torpor. As I said before, energy demands are reduced to the lowest possible level. Also, cell metabolism is altered in quite an intense way. For example, to supply the brain and the heart, glycolysis is shifted to lipolysis. Gene expression, protein turnover, and cell cycle are downregulated as well. And there is absolutely no digestion or nutrient resorption during torpor. And also the immune system is heavily downregulated. However, torpor has also some very shielding effects. So just keeping in mind how long the animals are inactive and lying around somewhere, there is no sign for muscle or bone wasting. The brain also, has no damage. There are some loss of synaptic contacts, but these are repaired during arousal, so it should be not be a problem. And it has been shown that cells from animals in torpor show a less susceptibility towards damage by reactive oxygen species, and they are also resistant to radiation. What we also know is, and this is now just focusing on mammals, is that the ability to perform torpor seems to be quite randomly separated between all mammals or between species of the mammalian kingdom. So it ranges from marsupials over insectivora, carnivora as bears, bats, some apes even, and rodents. And also the modes of torpor are very, different. One example is daily torpor. Daily torpor can be a seasonal behavior. However, it can be also induced quite artificially, and it can then happen as a facultative behavior in response to, for example, alterations of light conditions. When torpor lasts for several days, it is called hibernation. But even here, the features differ very strongly between the different animals that are able to perform or to be in hibernation. Just having a look here at the dormouse, a quite small rodent. It's able to reduce its metabolic rate to only 2% of its resting metabolic rate, and the lowest measured body temperature was at 0.7 degree, meaning that this animal was almost frozen. On the other side, we have the brown bear, a very huge animal. And here the metabolic rate drops to 25% approximately of the basal metabolic rate, and the decline in temperature is quite moderate with only 34 degree. So this was quite a superficial overview of what we know about torpor. But now it's going about what we do not know about torpor. So we don't know the torpor triggers and the torpor drivers, as Matteo also mentioned before. And in search of identifying this, several conditions were modulated in animals to induce torpor, like food shortage, changes in temperature, and also altering of light phases. Of course, several investigations have been also made on organs of torpid animals, like on the brain, heart, liver, kidney, to find something that is different between animals in torpor and their active counterparts. There have been already identified some substances like H2S, 5-AMP, and others that are regarded as putative torpor triggers. And of course, it is assumed that there must be actually some kind of genetic program that makes the animals able to perform torpor, but how it is activated or not, this is still a point of research. And this is where I come to our project, where we're investigating looking for the ability or the similarity of some responsible genes that trigger or drive torpor by blood omics. For this, we have mainly 2 different model organisms, as I showed you before, the brown bear and the Jungarian hamster. And we received from the brown bear blood samples from, luckily, the same animals in their active summer state and in the hibernating winter state. And for the hamsters, we were able to make a higher resolution of the torpid phases by investigating blood from animals at torpor entrance, mid-torpor arousal, and also after torpor, always in comparison to their normal, active counterparts. As Matteo summarized very nicely before, there are a lot of indicators or kind of evidence that torpor could be maybe also realized, or that is already a condition that can be performed in humans. And what he didn't mention is in his presentation is that a lot of evidence is also can be also found in very, very young humans, meaning newborn babies or even fetuses, because they show a very low metabolic rate, especially when they are still in the womb. That is much, much lower than it would be expected for a mammal of the same size. And for our analyses, we received blood from premature and mature born babies, of course from the umbilical cord vein, so everything fine. And also from babies 10 days after birth as some kind of control group. And with the material we got, we performed transcriptome, metabolome, and proteome analyses. So our studies are still ongoing, and I will give you now just a short status report where we are at the moment. So for the bear blood, next generation sequencing is completed, and currently bioinformatic analysis have started. For the hamsters, next generation sequencing as well as metabolomics and proteomics has been performed. The bioinformatic analysis is finished, and currently we are interpreting the results. What I can show you already now is that there is a lot going on the transcriptome level in the different torpor phases. And this is very pronounced, especially at time point 7, which is deep torpor. By comparing these changes at deep torpor with findings that were already reported in the literature, we have also seen that changes that go into the same direction show a independency of species, torpor mode, and even organ. Okay, let's come now to the babies. There we also performed on the blood sample next generation sequencing, metabolomics, and proteomics. The bioinformatic analyses are finished, and currently we try to interpret our results. And here's just a small insight what we found out by now. The gene expression analysis showed that the 3 different groups, so premature in blue, mature born babies in green, and day 10 old babies in red, can be clearly separated just according to their gene expression profile. And the differential gene expression analysis reveals, that is depicted here in the volcano plots, that when comparing the gene expression of premature born babies and mature born babies, there is a very, very huge number of genes that is differentially expressed in premature babies, which was much lower when comparing mature born babies with 10 days old babies, showing that the prematures are a very, very separated groups from the other one. Based on the differential gene expression analysis, we went then into 2 different directions, or we are planning to go into 2 different directions. The first is to identify which maturity state of maybe premature or mature resembles torpor the most.
And for this, we performed gene enrichment analysis with our differentially expressed genes. And we received a lot of biological processes that were altered, especially in the premature born babies. And I will show you here a short summary of what these analyses actually told us. So one of the most prominent points was that in premature born babies, the immune response was, or signaling pathways or pathways itself that were involved strongly in immune response and lipid metabolism were severely downregulated in the premature born babies. And then the expression of the relevant genes went up with maturity state. And comparing this with findings that have been made in torpid animals, the premature borns would match with the torpid animals. However, for lipid metabolism, it goes the other way around, since we heard before that lipid metabolism is strongly active in torpid animals. However, it's completely shut down or more or less shut down in premature born babies. So this didn't give us the big match. Okay, going the other way around, we have seen that especially genes involved in mechanisms of autophagy, glucose metabolism, and response to hypoxia were strongly upregulated in premature born babies, and this then declined with gestation age. And comparing again with facts we know from torpid animals, at least premature born babies would match quite nicely with regard to autophagy and response to hypoxia. However, glucose metabolism, as we heard, is shut down in torpid animals. So again, no real match. So we concluded that there is actually no real conclusion possible only by the transcriptomic analysis, and now we are complementing it by including our metabolome and proteome data. When this was then finished, it will be finished at one point, we're going to search for similarities of torpid mammals on the blood transcriptome level. We did with our literature research and the hamster samples, quite a comparable work before, but now we have the same sample material of the animals and also the same processing of the blood and also the same analysis method. So first, we are going to compare similarly regulated genes from the bears and the hamsters, and then we will go with our results to the baby data. And I hope that I will be able to give you a nice update in our next meeting. That's so far from my side. Of course, you know that this is a very, very complex topic, and good science is actually not possible by doing it alone. So I want to thank very much to Alexander Scheer and the whole stress and immunity team from the LMU hospital. Stefan Sim, who is our bioinformatician from the University of Greifswald, Dominique Singer, who provided us, so from the hospital in Hamburg, who provided us with the baby blood samples, Annika Herwig and Victoria Dietrich from Ulm University, who performed the animal experiments, Ole Fröbert, who provided us with the bear blood samples, and of course Jürgen Bereiter-Hahn, who initiated and organized the whole project. And of course to our funders and supporters, Uniciencia Foundation, ESA and DLR, and to the ESA topical team. Support. Thank you very much.
Thank you, Dominika, for this really great update. And now I have a pleasure to invite Dr. Andreini Pisari, so sorry. I hope I pronounced it well, but now I will call you Remy, to an update that I'm very much particularly interested in about like what is the radioprotective effect of hibernation. So Remy, the floor is yours, please.
Thank you very much, Anna, also for reorder. The presentation is much easier for me now to explain our research. Actually, I would like to request, is it possible to turn off only the front part, the lights, or not? Can I? because I have some immunofluorescence images that actually might be much easier to see because it's quite, yeah, if it's possible. But if not, I will just sort. Yeah, I think it's better. Yeah, thank you. So I would like to share our work that actually been done with Matteo as well and Walter Tinganelli from GSI about how to optimize synthetic torpor as the radiation shielding. So I would like to just introduce what is the risk of radiation exposure. Here I show you what happened to the brain. Actually, when we're giving irradiation, there is acute. Within 24 hours, the radiation can affect the behavior of the animal. And if we take a look more closer to the cells, to the neurons itself, the effect itself is not immediate, but it's gradually affected later on. Within 1 week, the neuron cells are decreased, and moreover, the synaptic function of the cells are decreased. And on the liver, so this is actually the studies done by Matteo and also Walter. So they irradiate the rats, whole body, and then they take liver of this rat. So I only show you this is the histology of the irradiated rats actually. So you can see that here the vacuolization of the cells occurs after the radiations. So what we can do to actually to mitigate these radiation effects. So one of the idea is, as mentioned already by Matteo and Dominic, is torpor or hibernations. It's originally also, the finding is in the 60s. There are studies on the natural hibernator and mice that when they do the radiation to these animals during hibernations, they're able to mitigate the radiations. So that was the start. And then the question is, of course, we are human, we are not able to go torpor, as like I already introduced before. So Matteo actually did induce synthetic torpor on rats, which is the same rodents like mice, but actually rats cannot go naturally into torpor. So what Matteo did is actually induce this torpor to the rats by injecting the muscimol, which is the GABA agonist, into raphe pallidus area. You can see here, it's shown that the rats, before the inductions, the body temperature is pretty warm. And then after a while, because of physiological changes, it's hard to see where is the rat because it's become same like the background temperature. And then in these conditions, together with Walter, they irradiate these rats and then collected the samples of the organs, and they saw that actually there is a radiation protection effects. However, this is quite invasive. So we are trying together to find out, could it be performed in a less invasive manner? And if we able to induce it, will we also have the same radiation protection effect? So it started actually on 2017. It's a nice paper from a team in India. So they used this adenosine agonist, these A1 receptors. So as just simple explanation, this chemical is actually the opposite of caffeine. So we like coffee, right? To make it awake. So this is really the competitive receptor. They bind really in the same receptors. So basically, if we don't have a caffeine, then this adenosine agonist will be binds to the same receptors. That's why we are making it feel sleepy, tired. So, and then they use this called 5-MP actually, which is bind to A1 receptors. They're able to bring a mouse in this study. They use black 6, this mouse into the hypometabolic state. They give irradiation first, and then they injected IP, so it's less invasive with 5-MP, and then they saw they can bring this body temperature of the animal is quite low. And then you see here they give radiations, and it's pretty busy, but with the 5-MP, This is the blue line, and they also actually use the radiation mitigator, these amphioxetins. Here, both have a better survival after the radiations. And after a while, it's also spare the white blood cells. And then they collect the femur, take a look in the bone marrow of the femur. Here, with the 5 MP and the low temperature, is preserved the bone marrow. And also the mononuclear cells here is being preserved even better with the radiation mitigator combined with the 5 MP. So that process was able to be done on mice. As I mentioned before, mice can go to torpor naturally. So how about on rats? So we did try, actually. This quite challenging was, we supposed to do, Mateo also come with us, and then the COVID came. We go to Japan to do this experiment using heavy ion in Gunma University. But COVID came, we managed to do the experiment. So what we did is we used the rats, we irradiate first, then we give the 5 MP IP. And then we put it in the cold room for 6 hours, and we measure the body temperature, and we collect the samples after 1 week. So in this time, we didn't manage to bring down the body temperature of the animal like 20 degrees, but we see that actually we lowered the body temperature of the rats. So what we able to achieve here is actually only moderate of hypothermia. So what happened to the organs then? So when we take a look on the brain samples, so upper panel is saline injections. The right side is the after carbon ions. You see this EBA1, this is the microglia, is the marker for the inflammation, is increased a lot. While with the 5 MP injection, the number is being suppressed, which means the inflammation is being suppressed. What is important is also if this microglia is really working as the macrophage, because they are working like basically like a vacuum cleaner. They have to clean the debris when this inflammation occurs. They have to actively active and also being functional. So we did the calculation between the active the positive cells for the macrophage and the microglia, and basically radiation actually suppress the functions a lot by the percentage, by the number percentage. Sorry. So it seems that synthetic hibernation can reduce the radiation-induced inflammation. And when we take a look on the liver, so previously by injecting mucimol, they saw that actually liver being protected after radiation with the synthetic torpor. Here, this is the saline injection. As you can see here, it's shown that, this is without irradiation, very nice, intact histology, while after irradiations, the sinusoidal is being dilated here, and also the nuclei is being, you see that some apoptotic nuclei. While with the administration of 5 MP, after radiation is comparable to the control. However, unfortunately, 5 MP is still drugs, it's chemicals, it's quite toxic. So if you see compared to non-irradiated, actually they're increasing also the cell death in the hepatocyte, but still less compared to only radiation without the drugs. So it seems that with this treatment, immediately after also can reduce the number of apoptosis and the cell damage. So this is what we saw on in vivo. We did check also with the cells in vitro, because we would like to see, understand what is either 5 AMP alone can reduce the cell damage, or we have to combine it with the low temperature and the low oxygens. So we did playing around with the oxygen concentrations and also with the temperature here. This is pretty confusing, but basically doesn't matter with the 5 AMP or the temperature alone, the DNA damage or the repair are being suppressed compared to the normal conditions. But what interesting is when we take a look more closer to the cells, if you recognize in here, this is the DAPI, the nucleus. After 24 hours, they do the cell death mechanism called mitotic catastrophe. The mitotic catastrophe is being reduced actually only with the presence of the 5-AMP. So that's what we have been seeing. It's quite promising, I have to say. But a lot of things that we have to answer. For example, can we do the prolonged torpor? And the toxicity consideration, as we saw already, the liver itself is not in the good conditions, and the nutritional status and effect of the radiation, other modalities. We did try, so this is still preliminary, prolonged torpor in mice. So you can see here, we use this 5-AMP and also CHA, also another A1 agonist. Here, CHA was pretty much, can bring the animal, goes to torpor pretty quickly, but it's pretty toxic that the animal was not survive after all. But then, we using 5-AMP, we manage, goes to 6 days, we play around also with the dose. And you can see, as showing here, is actually the animals were, before started, still warm. And then after a while, at 2 hours, I hope you can see also this shadow of the animals. It's actually quite close to the background temperature. And then some animals starting to be awake at 4 hours and 6 hours. And also some are still sleeping, actually. However, when we induce this every day, unfortunately, the weight of the animal is decreasing, which is not good. So we did modify it by giving it every other day, and we managed to maintain the body weight of the animals. But then after what we saw, you can see that actually some animals waking up very quickly, some goes really torpor very long. This is also still preliminary. It seems that the weight of the animals, it's quite related to the duration of the torpor. This is not statistically significant yet, because we have to add more animals, but this is something that actually need to be explored. Reason is because it's also the A1 receptor is more abundant in white cells, white fat cells. This is something that we have to explore, because the metabolism of each individual is different. Can some people have more white fat cells, can be more brown fat cells, which is then we have to modify which chemicals is actually work better for each individual. And this actually also showing on the ground squirrel during the hibernations and before, not during hibernating. This is normal, non-hibernating. This in bone marrow. This is AD, means adipocyte cells. When they are being not hibernating, the adipocyte cell is like that, but then when hibernating, the number and the size is also increased, which is something that we have to consider as well. And also, bear is going to torpor, and if you, I saw some documentary that they're giving milk, and the human trying. The taste is very awful, they said, because it's the fat content is very high for bears. I think they also being prepared for do the torpor during winter, so the percentage of the bear milk is 20-25%, while what we are drinking is 3 to 5%. That's in future. Also, that's what we did show is the base of the radiation of photon and carbon ions, heavy ions. My question is also actually, it would be nice to see if the effect to the protons. So since the beginning of this year, actually I moved from GSI to Holland Proton Therapy Center. This is a hospital, but we have also the experimental room. Well, my dream is that if we can do also the experiment here, because we have a really dedicated experimental room here, only for the radiobiology. which is very nice. Then we can do in weekdays, and it doesn't have to wait until very late night after the patient, because we have dedicated room here. And we have, of course, can do in vitro work with the GMOs, physics and chemistry lab. We have also the animal facility that actually during this mid-year, actually we managed to do the in vivo experiments. And this is, for example, well, this looks like a spacecraft, but for mice. So we can do a design actually for the holder for the animal for each experiment that we can use. And we can do accurately the dosimetry, because we always do it every week, even also every week, and also for before the experiments or during daytime is possible. And most importantly, we are currently also developing this incubator that can be fit in the beamline, because as you saw before, radiation very sensitive to temperature and oxygen. So if we can maintain this, we can mimicking more like in what's happening in our body. So that's very important, and then we can call it, because it's 21%, but actually in our bodies can be 4-5%. It's not really 21% of our organs. So this is what we are doing now. So our facility actually have been using for the harshness test already for ESA and for Rotnex. And as for the take-home message, we show already the effect of hibernation can be mitigate for the adverse effects for radiations, but still some questions need to be answered. And I would like to thank these people, because this work cannot be done with all the collaborations. And also thank you for the Holland PTC team. Thank you very much.
Thank you very much, Remy, for this very interesting presentation. So now please stay in the stage. And I would invite all the speakers to come to the stage with me. We would open Q&A right now, so if you have any questions, please raise your hand. However, I will set the stage, let's say, for the first question. And actually, first question to Matteo. I would like to, yes, I would like to, very interesting presentation. I would like to refer to the very beginning of your presentation. So at the beginning, you show on this experiment on mice that first comes slower metabolic rate and then temperature. Later on, you were mentioning about like different conditions have to appear. So I have a question to you, assuming that I'm in one of those 10% people and I want to do a sleepover, so winter over without climbing Mount Everest. What conditions would have to appear in which sequence for me to naturally go into this state? Do we know something about it, like the sequence, how to induce naturally in humans? Do you have any hints for that?
Right. So as Dominique showed the evolutionary tree, right? So there are random animals around the mammal kingdom, mammal world that enter torpor. The idea is that this is an ancestral trait. So first proto-mammal about 150 million years ago was able to enter torpor, coming from reptiles, and use it as a defense mechanism against predation from dinosaurs, and also to save energy in our very expensive body and high metabolism. When an animal can enter torpor only as a defense mechanism, if we see it as this, is called a facultative heterotherm, meaning mice don't enter torpor regularly, and it's not only mice, they do it if it's dangerous. And what is the danger? The danger is a negative energy balance. So if they see I have not enough resources to go on and keep the machine going, then I can enter torpor, saving energy, saving part of the day, and then I will arouse, see how conditions are, maybe things are changed. If they're not, I can enter torpor again. Let's put it in this way. Let's say I have 1 day left of life, and I'll decide, okay, if I go into torpor 12 hours a day, I can stretch my 24-hour life left in 48 hours and gain strategy to increase survival. To do that, that's to sense the negative energy balance, first has to come the increased energy demand. So your body needs to respond to an energy demand, which in case of Mount Everest, and not only for that, could be cold exposure. So, if you're cold need to use and pump up your energy metabolism to keep cool, to keep warm, sorry, and that your brain senses it and say, okay, I'm spending a lot. Then has to come the shortage of resources. Okay, I'm spending a lot and I don't have enough. And those are the two critical conditions. If you lower oxygen, it's not necessary for natural torpor, but we think it may give it a push to animal like us, which may not be really no hibernators truly in the main sense, because it will also send energy shortages even more severe because there is less oxygen. There was an interesting paper will come out soon about inducing torpor in mice, which, as I said, are in this condition facultative heterotherm, by using a diet supplementer, which is a drug, actually, it's not really a supplement, which prevent one of the futile cycle in the creatine and CHK energy production. And these mice enter torpor despite the fact that they feed. So they can eat, but they can really use the calories they assume, so they will enter torpor. So as I mentioned in sequence, you'll have to have a cold exposure, fasting, low oxygen, it might trigger you. But for a big animal like us, it will need a strong push. So a long fast and a very severe cold exposure.
All right, thank you. Very interesting. So I would like to follow up with. Okay, so we have a question before I get to mine. So please let me give you the microphone.
Thank you. Frank Posbeck, Freie Universität Berlin. Directly to that case of Mount Everest. So you have 2 out of 30, but this is in fact just a lower limit, right? It might have been possible that half of them or 90% of them entered the torpor state, but well, just weren't recognized soon enough before they died then, after all, after a few days. So only 2%, only 10% woke up, maybe even 30% woke up, but couldn't be recognized. So this is, what is basically making them wake up? So under these conditions, right? They are,
this is the deepest question in the field, because entering torpor is hard. I mean, we're now tracking the brain area that are active in torpid animals, so we know a little more about how to enter. We really know anything at all about how to come up. If you imagine an Arctic ground squirrel with a brain of -1 Celsius that wake up, what happened, how the biology start working again, that's a very deep mystery. Nobel Prize award if to either design, I think, in biology. So yeah, it's one of the limits, because also consider people going on Mount Everest are mostly male at the time, so most male than women, so harder to enter torpor. Good health, middle age to young people, so it's a selective part of the population, and we may definitely find more if we were looking among youngest on eldest and more female. So yeah, it's a conservative extreme.
Dieter Blottner, Charité Berlin. I have a simple question. We know from the clock genes that they are triggered by circadian rhythms, and just my simple question is torpor rather triggered by external or intrinsic factors?
It's an easy question for me. All right.
Okay, you win the Nobel Prize. But what's your idea?
So let's try to be quick. There are multiple kind of way to enter torpor. There are seasonal hibernators like bears, squirrels, bats. We think they have a circannual clock. Okay, so the gene expression that combine with the change in environment push into torpor. But if you put a squirrel in a lab and you don't allow him to see the outside world, keep the temperature constant, keep the light constant, in October, November, it will still try to enter hibernation. It won't quite make it as strong as is outside, so the bout will be shorter, you keep waking up, but it will try to go. So there is a genetic intrinsic factor, and those animals have perfected this evolutionary trait, so they have done a further step, we think. Then there are circadian regulated torpor, because daily torpor, like hamsters, they do it in the winter, but they do it daily, same time of the day, like the disease we've seen. And that's a combination of, as I said, cold exposure and clock originating. And then the facultative heterotherm, they do it pretty much only for external factor, as I mentioned. The environment put me in a negative energy balance, I'm responding, activating torpor. So we think the efferent arm is the same, is a network that goes from preoptic to dorsomedial hypothalamus, perigut total great, and raphe pallidus. But the stimuli that activate this pathway may be different, and they may have shape a different in terms of evolution. And we don't know which is our. We believe our is still there. We just think that animals like ours have lost the ability to activate it normally, and we could do it artificially. Of course.
So, Jan Rüttwiger from DLR. I have a comment first and then a question. So the comment is simple biochemistry, that any reaction will increase its speed with the temperature. So much of what you may be seeing in hibernation and waking up may be a positive feedback loop. So the controlling signals don't need to be very strong. There could be self-reinforcing mechanisms in it. And so when you have positive feedback loops, the question always arising is, where is the threshold for kicking this off? So, when you go to torpor, it may be the question, where does it start? And then that's related also to the question, where does it end? I like this idea of, cybernetic control, comparing the energy that you have and, that you can invest. Have you considered, and this is the question, that this comparator could be the equilibration of heat losses with the production of endothermal heat? And if so, then you've already mentioned the structures in the hypothalamus. They're very close, and that'd be an ideal comparator of investing what are my mechanisms that I invest for heat conservation and heat production.
Yeah, your comment is absolutely correct. We've been playing around with this idea. The point is that if it's the brain activating the phenotype, there is a theory that is out there. There is something like an hibernation hormone. So something that goes to every cell, go to the mitochondria, turn it off, and then you turn down metabolic rate and enter torpor. But that's never been really consistently proved. So common idea now is the brain activate the torpor phenotype. Question is how the brain signal goes from neurons in the hypothalamus to every cell in the body. Because at a certain point, the cell have to adapt to this new condition. So how's the order, how the command goes there? We don't know. But circadian biology is the idea I like to feed into this, to synchronize the clock genes within the body is also a problem. And the theory, correct me if I'm wrong, is that the SCN, the suprachiasmatic nucleo, sense the clock, is the master clock, decide what time it is, then tell the autonomic sympathetic nervous system to generate the circadian rhythm in temperature, and the other cell of the body sense the circadian rhythm of the temperature and synchronize the clock genes. So it's a gene to neurons to, let's say, autonomic nervous system to physics, because it's a temperature signal, to molecular biology again. And torpor I think, could be going the same way. So you have energy sensing structure in the brain, and which will have information from how's the periphery going and how's the environment going, and then say, okay, let's enter torpor. At this point, I tell the autonomic nervous system to initiate the mechanism in some tissue, in some specific part of the body, which will start lowering metabolic rate, and the rest of the body may feel a condition in which lowering temperature and reducing oxygen consumption. So maybe the gases in the blood, for instance, the PCO2 could be a signal, for instance, will be read as, hey, this is the command for enter torpor. I need to activate the genome part that tell me to enter torpor. So I would expect most of the bodies to, in terms of transcriptomic, to respond very similarly to torpor and hypothermia, and other organs to respond specifically to torpor in a different way from just temperature. So this is the frame I like to, let's say, interpolate energy sensing in the brain with synchronizing the biochemistry of every other cell. Because at the end of the day, mitochondria will have to turn off, but they will be able to turn back on command. If you have a mitochondrial inhibitor, that stays there, you're never going to wake up as a cyanide. So mitochondria have to lower their activity, but be ready to start again. And that's only an instruction that can come from, let's say, something as like as the circadian rhythm regulation.
Thank you very much. And before I get to the next person that would like to ask questions from the audience, I would like to shift a bit the discussion maybe, because we heard a lot about the molecular effects. So I would have the next question to Dominique. So having what we know about the molecular effects, and you are studying molecular effects, we were talking about the natural induction. Is it possible to hit a specific pathway already? Do you have hints there to actually induce it synthetically?
Unfortunately, by now, we didn't see in our analysis if there is really one specific pathway that can be hit, because it's not like that you can, direct one gene only to one specific pathway. It's that they are involved, or a lot of them are involved in so many specific ones, so that there is still a lot of research by now to do to get there more insights on this.
Hi, Thomas Bode from the Charité in Berlin. I love the concept, fantastic talks, and I love the broad difference between the basic science, the mechanistic science, the concepts that you are presenting. However, I feel like there's a massive delta, right? So we're doing molecular science, that's great. And on the other hand, we have anecdotal evidence from Mount Everest. Now, I think if we ever want to fly that actually in space in the next, let's say, 50 years, we would need to leapfrog at some point. So I have a question, probably it's a good reason for not doing that, but would it be an option to just take 20 chimps, 20 pigs, whatever, maybe not fly them to Mount Everest, but put them in a climate chamber? And maybe if the hypothesis is right, 10 or 20% of them will enter torpor. And then also run all of your metabolomics, all of your biochemistry assays on all of the animals and see what's the difference between the ones that enter torpor and the ones that don't enter torpor. And if none of them enter, then then we have an issue. You have an issue, I guess. But but if some of them enter torpor and you see a difference in the biochemistry, then you have an actual target to work on. And I think that could close the gap between the concept and the anecdotes to the actual fundamental science.
Yes, that's we've been playing around that. So doing the animal experiment you're suggesting will face a very hard wall to break in the term of the ethical committee. So and not even talk about doing it in humans. Absolutely, that's why Mount Everest was an interesting, natural event that could be interpreted. To clarify a little bit more in terms of biochemistry and also genetics. So far, there is no clear indication that there is the gene of hibernation, the biochemistry of hibernation. There is nothing really different from an hibernators to non-hibernators. It's probably just the way the genes work together instead of I have this special protein that you don't have. Maybe with some exception, the heart may have some calcium channel in hibernators that are slightly different, but there's very details. So everyone has tried to do kind of what you're consistent. So proteomics and transcriptomics and hibernators versus non-hibernators versus animal we send into what we call synthetic torpor. Rainey have shown you can do it, and with adenosine-derived, let's say, drugs. You can also do it by selectively inhibit or activate brain area, so only there, so no drugs around. The response is there is nothing really specific. There is just the phenotype adaptation of the system. So it's a little harder than what we think. Consider to do what you're considering for sleep. It's kind of the same. There's not the genes of sleep. There's no sleep pattern where when you sleep the brain is different than when you don't sleep. It works differently, and understanding what sleep does is indeed another big question in neuroscience. Dominic, if you want to add something.
I just can fully agree that we have to search for specific patterns, and here especially for all the omic analysis, it's a very different point to say or to set then the thresholds, what genes or what substances are really then worth to have a closer look at, because to set here the significance point is very, very hard. But I also agree it's more another way of working together. And we have to look for specific patterns, and I don't think that we will find the main inducer that you can really define it as one specific gene or protein.
just quick addendum. We're more investing now in how to mimic it, not necessarily how to replicate it from the start. Like, if you want to fly, you build an airplane, but the airplane doesn't flap the wings like birds. So, but it still flies. So, understanding how it works in the natural kingdom, could we infer a way to hack the system in non-hibernators? This is more the concept we are discussing about now.
Okay, yes. Maverick Kuipers from ESA, flight surgeon and also emergency physician. Wonderful presentation, thank you. I have a couple of thoughts, and I think you already hinted at it. So we actually, humans, go into a daily mini torpor, right? We go to sleep and our body temperature drops. So I think, are you working with specialists in the sleep field to look for these patterns and synergies? And then I'll continue with my other question.
Okay. Well, brief answer, yes. There is a theory that the sleep torpor hibernation, because a big continuum of how heavy is your metabolic suppression. It's been debated anyhow how sleep and torpor parented are similar, but we work on that also for a different angle, which I haven't discussed at all. That when animals wake up from torpor, we mentioned sort of why they wake up. Well, if we look at what they do when they wake up, they sleep. So the idea is that during torpor you don't sleep, and so you're sleep deprived. And sleep drive may be one of the forces. We don't know what sleep does, but whatever it does is not done, and therefore trigger the arousal. You need to sleep. You don't need to feed. You need to sleep, and then you can enter torpor again once you have slept. So there is definitely a deep interconnection between the two behavior. Even harder to study the two separately, but torpor hibernation can give you a window on sleep research that is very original and very different, as I mentioned, because you're sleep deprived for a long time, and that's open a different angle on investigation.
Thank you. Yes. And so my follow-up question, so in clinical medicine, so patients with sepsis or severe trauma, a lot of these protective effects can be very useful as well. So I think there could be an entry point for human-based research, because if we are, if we have humans that are to a near-death situation, and you want to kind of push the stop button and keep a pause and try to fix things, that could be an entry point where we could actually see where the effects could be in humans. So is any science directed in that direction?
You're absolutely right. Is as today, we could replicate what we've done in rodents in human with the high risk that this could kill the person. So, is at a certain point we'll have to what the path that we're thinking is we need to strengthen the, let's say, the translational basic research, the animal models, as a certain point at which we consider safe enough to risk it, and the risk has to be taken in no hope conditions. Something for we were considering hyperacute liver failure, like your liver died and you have 1 week to live eventually without liver maybe, and at this point if you can spread this week into a month, maybe the chance you find a donor increase significantly, and maybe you can be bridged back to life. But septic shock is another possibility. It's just this valley that you have to cross, where who is the first one who does that, and it's going to be something invasive, like we go in the brain and do something which is increased as a risk intrinsic to the procedure. Neurosurgeon told me, no, I don't care, I can inject whatever I want, whatever I like, but truly there is a risk in the procedure. And or pharmaceutical, like systemic drugs, or a combination of the two. We would need, I would say, a strong body who really willing to do it, and then do this critical step. But if we fails, the entire field is doomed. So we prefer to work with care.
Thank you very much. And because we are running out of time, last question from the audience, and the rest I will have to ask you to bring to the inspiring discussion in coffee breaks, please.
Just to end with a spiritual ending. So I'm from an Indian background. I have heard stories about yogis who go into this kind of meditative state where they can lower their metabolic rate so long and they can stay in the same pose for a longer time. Have you ever investigated? Because I've never seen them, but I've heard from that the breathing techniques and these things could really do that. Have you ever? I would pose to all the three to answer this. Thank you.
Thank you. We indeed had a very experienced Qigong master of meditation in our lab, and we followed his meditation process really from the beginning. That was very arbitrary, so he prepared himself with regards to breathing and really deep meditation, and it was so fascinating to see because he then lay actually in our lab and he was completely like knocked out. He was completely gone. And we also took a lot of samples regarding blood, also for transcriptomics, breathe, ECG, everything we were recorded during this time. Unfortunately, we are not now by then to have it fully analyzed, but we investigated such a Qigong master, and it is a very fascinating process, and it shows again that this is an active process. It is not something which happens passively, definitely.
Thank you very much. I will finish with the question to Remy. Remy, because we didn't touch in the discussion upon the radiation protection effect, which I believe, of course, I'm biased, that is the most significant function of hibernation. So would you please, giving the spaceflight scenario, would you please say, is there like new updates in knowledge on the effects on how effective torpor is towards the heavy ion component of the galactic cosmic spectrum? So are there differences between the low LET and high LET radiation effects regarding torpor? What do we know? How optimistic it looks for the radioprotective effect in spaceflight?
What I could explain is actually based on the studies that actually Matteo did with the X-rays, with photon, and what also we did also with heavy ions, with carbon ions. We see the protective effect for both. It's just, that's why I think it's going to be interesting more in space is there are mixed ions. That's something that actually we need to test it. I hope in GSI they will have this mixed ions in 2028, I think Marco was mentioning, if I remember correctly.
Actually, already next year.
Okay. That's something that I think we have to test it there. And also for protons, this is my own ambitions. I hope, Matteo also and the other researcher also still interested to work together, because a lot of questions need to be answered here. For protons, for example, of course the astronaut will go to passing through the Van Allen belt. It's a lot of protons, but it's very quick. I know. I mean, it's like 30 minutes. But if nowadays the astronauts are actually going back and forth more often, if I understand correctly, it's not like a long time ago, which is being exposed also to protons. That's something that I think is worth to explore more. Yeah, that's the thing.
Super, thank you so much. Thank you, Rami, Dominique, Matteo, for your inspiring presentations. Thank you very much for bearing with us, for your patience, and we are looking forward to the discussions in Couloir. Enjoy your lunch. Thank you.
Thank you, Anna.